But I think it would be helpful for him before he talks about himself and also about the Botanisol analytics solution. It would be really great if we could hear from you and from your colleagues, who they are, where they are, and what their activities are.
Okay, so Philippe, perhaps you could start to introduce the group.
Sure. Can you hear me? Yes?
Yes,
we can hear you.
So, Philippe Moingin, I'm an immunologist, and I'm actually applying artificial intelligence to design and develop new therapies for autoimmune diseases. I created this working group 2 years ago. We're roughly 25 people from the academy. I send you, David, the list actually of participants and or fields of interest as it relates to artificial intelligence. Irène, tu veux te présenter? Enfin, il te connaît. Oui.
Yes, David, you don't know me, but I am a foreign correspondent for Israel to the French Academy of Pharmacy. I am the chairman of ERANIM, which is a society for the Israeli Society for Medication and Vaccine Safety. And I'm organizing a conference within 1 year about the lessons from COVID. So,
and Irène, more importantly, Irène is my great neighbor as well here in Jerusalem.
Okay.
And friend.
Yes.
Impressive. Very impressive.
Okay.
Okay.
Could you go on introducing yourself, because we are a very small number of people, so everyone could introduce himself.
Marc, you want to introduce yourself?
Yes. I am Marc Vaz. I am the head of the biology department from a general hospital near Paris, and I am working on COVID-19, of course, and we are looking to try to improve the diagnosis and the prognosis of on biological parameters. So,
okay, thank you. We have then Pierre.
Yes, I'm now a retired industrial pharmacist. I had a very long career in the various drug companies in France, and some of them international, some of them French. My background is a pharmacist. I used to work mainly in quality issues and regulatory affairs. But now I'm retired and particularly interested in this field of artificial intelligence, but I'm not a specialist. But interested. A member of this group from the beginning.
Thank you, Pierre. Philippe?
Hello, I'm a biologist, a medical biologist in the middle of France in Clermont-Ferrand. I have the same work with Marc Vaz. My English is very, very poor, so...
we can understand you without any problem. So that's the people from the academy we have now. Some others will connect down the line. So you want to introduce you guys on your side?
Yes, I mean, I'll just do a quick one for myself. And, I'm David Litoff, and I'm doing a lot of work in the area. My background is actually in finance, CPA, and did many years as a CFO. But I'm also now doing a lot of work in business development and connections between companies. I have a company in Japan, which we connect to Israeli technology companies. And that's where I came across Tatum and Ari Goodman, who were very kind to introduce me to Dave Talenfeld and to what he's doing. And when I heard what he was doing, I thought it was a great idea to see how I could help promote this tremendous solution, which I was very much impressed with. Again, Dave is going to talk about the solution. We're not trying to sell anything. This is not a sales call. This is more, you guys are mainly medical doctors. We want to talk to you about it.
Commercial activity.
No, it's not commercial. This is more about just telling you and exposing you to this new solution that we hope will be implemented around the world. Dave can tell you a little bit about the clinical trials that will be going on in the US and what they're doing over there. But we wanted you to be aware of this as well. So you're probably one of the first ones. We have people in Israel that are working on it. We have people in the UK, and now we're happy to bring in many members, distinguished members from France, that we can introduce this solution to. So Ari and Tatum, if you want to just give a quick word, and then we'll hand it over to Dave, who will do most of the talking today.
Absolutely. Firstly, it's a pleasure to meet all of you. My background is intellectual property law and commercial law. And this brings us into creating strategic partner relationships, license deals, investments for clients that are raising capital through various different opportunities. And I have around 25 years experience of this work. So I'm very pleased to have be on this call and to meet you all, such great academia people. So that's just in a quick nutshell. We work globally with various different clients all over the world, as well as all the people that we create our partnerships with and so on.
Thank you.
Well, thanks everyone for making this connection. I'm Dave Talenfeld. I'm the CEO of Botanisol Analytics. I'm a serial life science entrepreneur. I am still in the process of developing one drug product, a novel anti-inflammatory, which is a possible substitute for drugs like Humira with fewer side effects. And it comes from a botanical source, which is where the Botanisol name of our family of companies comes from. So we've got a patent application on a small molecule that's very exciting there. I moved on to start another company manufacturing drugs and other ingredients from botanical sources, and that company was sold in 2020 to a publicly traded firm. And this is my third company, and this is an outgrowth of about 2 years that I spent directing due diligence on medical device investments for what was at that time the leading medical device fund in the Southwestern United States. So I'm thrilled to be here to tell you about what we're working on and how it may be relevant to you in the future, and also to hopefully make some connections that may be fruitful in the future for research collaborations and otherwise. Would be thrilled to have a reason to go to Paris and take my wife there. So please give me a reason if you can.
We'll try to do so. Thank you.
Wonderful. Okay, so I'm going to share with you a presentation that I prepared for the National Institutes of Health here in the United States. We just went through a 6-week program with the NIH. They are the largest funder of medical research in the world. And I am currently, today, you have a special treat because I'm at Johns Hopkins University, which is the largest recipient of NIH funding. So you'll actually get a chance to see the machine in action. And I'll show you how simple it is to program it to detect a new disease, and then to test for it, and to transmit the ability to detect a new disease to any location in the world, so you can immediately begin testing. So in terms of the technology, and I'll share my screen, there are a whole lot of advantages of this. So we can train for a new disease in under a day. We can then push that out as a software update to be able to detect it anywhere in the world the following day. So contrast that with the traditional way of doing things, which is, let's take coronavirus as an example. Here in the US, it was about 9 months from the start of the pandemic until the point where we had adequate PCR resources to test everyone. And with this technology, potentially when a new biological agent appears, we can characterize it in a day, push it out to all of our machines worldwide, and immediately be testing a potentially unlimited number of people for it. Each laser test takes only 6 seconds. So that's about 150 times faster than current state of the art. So you can imagine extremely high throughput. You can test many, many people per machine per day. And it's also very cost effective. The per test cost right now is about $2, at least our cost of goods, which is about 10 times better in terms of cost than the next best test. So tremendous advantages both for medicine and also for biosecurity, for national security, and happy to engage with those stakeholders in France as well. So this presentation, the NIH, we make two versions of the machine. One version is a small box, and I'll show that to you shortly. It's about this big. It's hand carryable, and you can set it up anywhere and test. And version 2 that we're working on is a sort of like an airport check-in style kiosk that can be self-service for testing in the future. And we hope to put this in retail pharmacies as a primary point of departure for medical triage in the future. In the United States, there is a retail pharmacy within 5 minutes of 90% of the population. So nearly everyone in the US has access to a CVS, Walgreens, or Rite Aid. And they are further from urgent care centers, and those are significantly more expensive. So imagine a kiosk at a pharmacy open 24 hours a day, within 5 minutes of nearly every American, where they can go if they're not feeling well and find out what they're suffering from, and then potentially have their medication dispensed at the pharmacy at the same time. Our company was founded in 2017, so we've been at this for almost 6 years now. We were one of the top 5% of applicant companies accepted into a program called the Air Force Accelerator, powered by Techstars. And our early funding came from the United States Air Force, from their Air Force Research Laboratory Biosurveillance Unit, and also from the Air National Guard. And the initial task was surveillance of new diseases and also protection of key personnel and critical infrastructure in the event of pandemics. We actually started work for the Air Force Research Laboratory in 2019, about 6 months before the pandemic struck. And we were immediately offered additional funding when the pandemic happened in order to try to accelerate the project. We've won 2 Air Force contracts of about $2.5 million. We've raised about $4.1 million from private investors. And we have now tested thousands of patients. We tested about 2,400 patients just to optimize the system alone, just to optimize the hardware and the test cartridges. We followed by performing clinical validations and a clinical study. 104 patients were tested at Harvard Beth Israel Deaconess Medical Center. 203 patients were tested at a field test in Windhoek, Namibia. And I'm now at Hopkins today, Johns Hopkins, and we are running about just over 300 patients here. So we have consistently seen very high performance, 97% plus accuracy in diagnosing coronavirus. And we are now adding, right now we are adding the flu to the capabilities of the machine. We've tested it on multiple sample types, 3 different kinds of virus transfer media. We've tested it on genetically and economically diverse populations, from rich white people in Boston to much more genetically diverse and less economically privileged folks in Africa. We've tested it in populations that have a high rate of confounding diseases, of other diseases present, and we've also tested it in folks who, probably only have one or both of coronavirus and flu. And our performance is consistently in the high 90s across the board, so it's very exciting. Next, we plan to test for saliva, and saliva is an ideal sample type because everyone has it. There's no supply chain of virus transfer media necessary. It can be as simple as licking the test cartridge and inserting it in the machine. And it's also a transparent medium, which helps when the laser propagates through the sample. There's nothing to obstruct it. optically.
Dave, before we continue, how do you want to, Irene and Phil, how do you want to handle questions? Should we, every so often, would you like Dave to stop for questions, or do you want to let him get through most of the presentation and then ask questions?
I think it's really your call. I mean, Dave, it's really as you feel most comfortable, really.
I think I'm comfortable, Dave, probably, to have a conversation. So
Yeah.
if you feel that you have a question, Dave Talenfeld will not be insulted if you asked it during his presentation. Okay.
Okay, then we can start.
So maybe now, if there's anything until now that you want to ask, please go ahead and ask.
On my side, I have a question. So you said, Dave, that saliva, I understand, is your preferred sample. This means that your tests will be then qualitative and not quantitative, right? The point being that depending upon the how fluid, how much liquid is there in the saliva of the people.
So no, it is quantitative.
Okay.
And it also is capable of accommodating a wide variety of sample sizes. So it's quantitative because when the laser scans the sample, it produces a two-dimensional spectrum. On one side is the wavelength of light that's being returned, and on the other side is the intensity of the light, the amount of light that's coming back at the sensor at that wavelength. So different wavelengths correspond to different types of molecules in the sample. And so you can actually quantify them, because you might see molecule X is only at an intensity of 100 counts, and molecule Y is at an intensity of 1,000.
Okay.
So you can actually estimate how much virus is in the patient.
Okay.
And we believe we're able to do that at concentrations as low as 100 copies per milliliter.
Okay, I guess you're going to. I guess you're going to illustrate or to tell us about how it works, basically.
Sure, and I also, in terms of sample volumes, we typically use about 60 microliters of sample per test. However, we've done it with all different volumes, and we've even tried it. Instead of using a micropipette, we tried it with a sort of a medicine dropper or an eye dropper, just to see if it would throw things off. And it turns out it doesn't. And this is advantageous for our military users because we want to make it as foolproof as possible. Use it anywhere, don't need to be a medical professional, and can even use a medicine dropper and take saliva. So that's very exciting.
Maybe if I can, come with another question. Have you tried, as well, to look into blood, just by pricking the tip of a finger, for example, in order to detect, say, pathogens which you might not necessarily find in the saliva?
Yes, we should be able to do it. We've not done it yet. We started with nose swabs and saliva because it's much easier to do.
Sure.
And less legal liability for us in the U.S. But we do have another project pending at potentially at Mass General Hospital in Boston, also a Harvard hospital, and they would like us to look at markers for both traumatic brain injury and post-traumatic stress disorder in blood. So that's something we look forward to verifying hopefully in the coming year.
Okay.
We've won a lot of awards. Some of these are visible on the website. Intelligence and National Security Alliance Award, Startup of the Year. We spun out of the University of Arizona, and they gave us a Startup of the Year award. The Flynn Foundation selected us for Bioscience Entrepreneur of the Year. The Armed Forces Communications and Electronics Association selected me as a top 40 under 40 industry leaders, and many, many more. Some of these are viewable on the website. I think we already covered this. Raised about 2.5 million from the Air Force and 4.1 million from private sources. We now have investors in 10 U.S. states. Canada, India, Singapore, and the United Arab Emirates. We also have access through a very unusual Air Force program, a brand new program, to up to $15 million in matching funds to continue to pursue this technology over the next 4 years. We spoke about the pharmacy kiosk concept, and on a high level, the advantages of this test over current state of the art it's multiplex. The same laser spectrum can be used to detect a potentially unlimited range of diseases. It's instant. We're able to perform the test in as little as 6 seconds, and it's cheap. Our cost to manufacture each test cartridge right now is about $2, and that can come down to potentially something like 15 cents at mass production scale. We also have the ability with this test not only to learn to characterize a new threat and be able to deploy a new medical diagnostic test in as little as 24 hours, but we can also use it, because it's a networked technology, we can also use it for biosurveillance and for public health applications, monitoring where outbreaks are occurring and making inferences about new outbreaks. The technology, how does it work? We use a technique called surface-enhanced Raman spectroscopy. A Raman spectroscopy is named after Indian Nobel laureate Chandrasekhar Raman, who was the first to observe that when light passes through a sample, some of that light interacts with and excites the molecules in the sample. And it causes them to vibrate and emit back a slightly different light that is unique to each molecule. So every molecule has a wavelength of light that comes back that is as unique to that molecule as a fingerprint is to a person. And so by observing this phenomenon, we can tell which molecules are in the sample and also quantify them. Are there any questions about that?
No, that's
I
yeah.
just wanted to ask if it's only about infectious pathogens or it's also for other disease.
Well, we've so far focused on infectious disease, but we're very excited to look at other diseases. For instance, cancer markers are.
So you say actually, yeah, biomolecules. In fact, right? That's anything that I guess any molecule which has a pretty decent size, I presume. Can we say that?
That's an excellent question. I don't have an indication yet that it has to be a large molecule versus a small one. So again, more research needed, but so far we don't have an indication that the size matters.
Okay. Interesting.
Okay, great. We talked a bit about this. The laser strikes the sample, it produces that two-dimensional spectrum that you see on the right. It's a little bit like when you hold a glass prism up to sunlight and it breaks the white light out into a rainbow of colors. And this is a technique that's used in astronomy to detect the composition of, let's say, gases on an Earth-like planet. And it can also be used biologically. It has historically been used for chemical analysis, but several recent improvements in the technology have enabled it to be used for biology with very high precision. Some of those improvements include thermoelectrically cooled sensors, which don't require cryogenic gases to achieve very high sensitivity with very low noise. Also, lasers, narrow line width lasers have been developed that make it much easier to differentiate between the laser light itself and the return signal. Machine learning methods have improved significantly, so there have been folks trying to use surface-enhanced Raman for disease detection for maybe 30 years. It's recently come of age in terms of the machine learning methods. And then also, crucially, the nanoparticle substrates are necessary to boost the signal of something very small present in the sample. So there's a phenomenon called surface plasmon resonance. And basically what that means is that when you apply very small metallic particles to the, let's say, the microscope slide that you're placing the sample on, when the laser strikes those molecules, it interacts with them in a strange way that actually increases the signature, the measurable signature of the molecules in the sample. And it's a strange thing that, you know, it is difficult to explain, but it works very, very well. And we have invented and patented a new way to make those nanoparticles that is extremely cost effective and repeatable. And that's one of our key enabling innovations. We've tested nanoparticles from all over the world, Norway, Japan, all parts of the United States, and others. And we found that the existing nanoparticles weren't really up to the task. So we invented our own, and we used about 2,400 patients to optimize our own nanoparticle substrates that make this possible. You really can't do it with anything else that's commercially available, and we know because we tried. This technique, I will also say, it's similar. We do have examples of machine learning methods which have been approved in the United States for medical diagnostic applications, and I would compare it to cancer detection in radiology. So MRI and X-ray images are now routinely analyzed using machine learning to diagnose cancer. And we are doing a similar thing, except instead of looking at MRIs and trying to identify tumors, we're looking at two-dimensional graphical spectra and identifying infectious disease states.
So the machine learning here consists, if I understand correctly, in recognizing a spectrum associated with a given biomolecule as a feature when you have a complex mix of molecules within saliva, basically. Am I correct?
Yeah, and one thing that's different about the machine learning approach that is sometimes hard for traditional molecular diagnostics professionals to grasp is that the machine learning doesn't just consider, when you use a PCR test, polymerase chain reaction, you're looking for a specific, DNA or RNA fragment. And you know exactly what that fragment is and that it's specific to this disease. And you look for only that one thing. And if it's there, the person is sick. And if it's not there, they're healthy. Machine learning is, we don't instruct it. We don't tell the machine what to look for. We feed it lots and lots of data, and we ask the machine, what are the molecular artifacts that are associated with this particular disease? And those could be, DNA fragments as recognized by PCR, but it also could be proteins, antibodies, immune responses. So it considers all of them, and it identifies the molecular artifacts that it determines are always there in a sick person with a given disease and are not there in a healthy one. But we don't have to tell it what to look for, nor ultimately do we really care much what those artifacts are. As long as the performance of the machine is consistent, it consistently and accurately diagnoses the various diseases. We don't have to know what it is detecting. We just lose.
Yeah, at one point though, I guess you've been interacting with regulatory agencies, with the FDA, for example. Of course, there's all this interest about transparent AI and the fact that, you want the algorithm or the machine to tell, what it is looking at. And I think that for regulatory acceptance of your technology, you may have to come with a rationale with respect to which molecules you're associating with the disease diagnosis, I presume, huh? No?
Well, it depends on who you speak with. It's a radically new paradigm, machine learning for diagnosis.
Very clear, yeah.
And some people are frustrated by the concept that we don't have a particular target that we're detecting. But there are great advantages of not having to name a specific target. And to some extent, I think this is a generational issue. If you're above a certain age, you're very skeptical of machine learning. It seems to be often the case. If you come from a traditional medical diagnostic background, there is skepticism. But if you look at tools that are emerging like ChatGPT, we don't tell ChatGPT to study a given legal text. We feed it all the data that is available. But we do know that ChatGPT can now pass the bar examination for a law degree in the United States. So,
Yeah,
it's a radical change. And one thing we can do for regulators that are curious about what are the things we're detecting, we can program the technology to detect a given disease, and then we can use contrived samples to better understand what is it that it's detecting. So, for instance, we can spike samples with different proteins or DNA fragments, and we can affirmatively say, yes, it's detecting that particular thing or not. However, in the long term, slowing the technology down is not in our humanitarian best interest. So if you want to identify the specific things it's detecting, it can be done. It's a time-consuming process. And the advantage of the technology is we can characterize a new disease and be able to detect it worldwide within 24 hours. So we expect that the regulatory paradigm, we may have to initially be very specific about what we're detecting, but as we move on, we expect that if the benefits to humanity are to be realized, then the regulatory paradigm will also have to change.
Sure. No, I think that's fair, but still, I mean, to receive your I would say your first certification, and I don't know at which stage you are, you will have to go across the could I say the acid test, I mean, to prove that indeed you detect in a very specific manner a given disease. So meaning that you will have to compare, I presume, with conventional diagnostic approaches, I guess, huh? I don't know, you will tell us.
I think that's likely to be the case, and I think in that instance it'll just involve a lot of contrived samples to make those determinations.
Okay.
I would like to understand something. Do you have like a library of a lot of biomolecules that the machine is comparing to what they are receiving their samples, meaning that how the machine is knowing that it's COVID or flu or cholera or whatever, or smallpox or whatever. So how do you do that? I understand that the machine is screening everything. So how the machine is making the diagnosis of a specific disease? This is what I've not completely understood.
So that would be the case in traditional Raman spectroscopy, or really any kind of spectroscopy for that matter. You would generate a library of known analytes, and then you would look for those analytes in the sample to determine whether or not they're present. In this case, we don't create a library of analytes. We create a library of what we call classifiers. So a classifier is an algorithm that has been optimized for a specific disease. So we have a library of disease algorithms and algorithms for different sample types. But we don't maintain a molecular library. So we don't have a library of proteins, for example. We have a library of algorithms that are unique to different sample types and diseases.
Yes, I have a question. So can you identify new pathogens, for example, even if you don't know exactly what is it, but could you have discovered SARS-CoV-2 or with your machine indicating there is an abnormal signal associated with clinical data?
Sure. Yeah, we're doing it. We've now done COVID and flu in multiple sample types successfully. And that is one of the advantages of using a machine learning approach, is that if there are anomalies, you can identify them. So for instance, if you know how to detect the flu, you may be monitoring for the flu, but you may see something that is not the flu, but is similar enough to flu that you can make an inference that this is, some other flu-like disease, and then you can characterize that disease.
Yeah.
So yes, that's something we're doing.
Are you able also to see the different variants?
A great question. We've not tested for it yet. We do have a patented technology that is designed specifically to do that. We haven't tested it yet. So my answer is, I believe so, and we'll try to confirm that sometime soon. Okay, and in general, this surface-enhanced Raman spectroscopy for diagnosing disease has actually been verified to perform very accurately many times with various diseases at universities all over the world, from infectious disease to cancer. However, it's typically been, I mean, you can do a Google Scholar search and you'll find tens of thousands of papers that have been written on this over the last 30 years. And, you know, probably hundreds of them that have been written in the last, 3 years that pertain specifically to coronavirus. This has been done many times successfully in research laboratories at various universities. So it's not in doubt whether the technique works. And really what we've done is made it work repeatedly, reliably in a smaller, more cost-effective, more robust way that can be deployed outside of laboratories. We talked about this a little bit, things that we've done to make that possible. So for instance, when it's typically done in a laboratory, it's a very large spectrometer that includes a microscope. So the signature is actually characterized by a microscope on a very small scale. One of the advantages of our nanoparticle test cartridges that we've developed and patented is that you no longer need a microscope. You can have a very large, excuse me, please. We're in the meeting. Sounds and just be aware you're on camera if you want by. Please mute the audio. Thank you. So one of the advantages of our new patented innovation in surface enhancement particles is that you no longer need a microscope. You can use a standard laser probe with a very large diameter spot size that you're illuminating, and you can build that into a very small portable machine, which I'll show you in a moment. And it works very, very well because the homogeneity of our particles is excellent. And in that spot size, you get a very nice distribution of all particle sizes and morphologies. So we've taken it from something that is very complex and requires expensive, sophisticated equipment and turned it into a small, portable, hand-carryable device. And that surface we've developed is a key enabler. The equipment is also very robust. So we built this for the military, and we have laser life estimated at about 10,000 hours of continuous operation. We have solid state components that are mounted into an aluminum hard case. And we have had these systems at military live fire demonstrations, on the backs of military vehicles, in the rain, in the mud, and transported to 5 different countries. And we've never had one go out of calibration. When we use them in the field, we can also cause them to self-calibrate prior to each patient sample, and that's a software feature for the future. And if you're curious, we can share this. So this is an example of the system on the back of a military vehicle, and we can just sort of zoom to the relevant. Here we go. So you can see it being used on the back of a Humvee. We have special forces personnel here. And we're able, in this case, to use a medicine dropper, collect the saliva sample.
Oh, I completely missed.
And I'm in a hazardous materials suit here in the North Carolina summer, which is very hot and humid. And I was terrified during this demonstration that I was going to faint in front of the tough guys. So you can see here we've inserted the sample. We've just clicked scan. The software application is very, it's a single button interface. It's very difficult to mess up. And we're able in real time to produce an assessment here and then notify, in this case, the military user that they've not been infected with a biological agent in the theater of combat. Going back to the PowerPoint presentation. So the equipment is very, very robust. Here's a comparison with current technologies. We spoke about this a little bit. We have accuracy that is comparable to polymerase chain reaction, except that we're about 150 times faster than the next fastest test and about 10 times cheaper per test than the next fastest test. We spoke about the intellectual property a little bit. And in terms of our research collaborations to date, we have worked with the Air Force Research Laboratory, Harvard, Rensselaer Polytechnic, and the University of Arizona, and as well as Lady Pohamba Hospital in Windhoek, Namibia, and now, as of this week, Johns Hopkins University. This is what the current machine looks like, inside and outside. I'm going to show this to you in a minute, so we don't need to dwell on this slide. And this is, again, another video of the current system in use. You can see you just insert the microscope slide, turn the system on, and you enter the patient name or number, and it begins scanning. Here we go. And this is our pharmacy use case prototype. It's a little bit like a check-in kiosk at an airport or an ATM machine. So we call this version the ATK, the autonomous testing kiosk, much as a cash teller machine is called an automatic teller machine. And this is something that we are collaborating with the NIH to define the parameters of what would have to be proven about this machine in order to use it, to have lay people use it on themselves, with all the considerations of sanitation and safety and contamination and consistency of sample type sizes, etc. So we're working through those issues, and we think this is the future. And fortunately, so far the NIH has agreed with us that they would like to see this technology fielded. So we're very excited about this. Another possible use case is airports. For example, transportation safety. If you've got a new pandemic spreading in the world, you might want to be able to restrict infected people from entering your country. So that's another possible application.
Excuse me, how do you handle the risk of contamination between different samples, different patients?
Yeah, so great question. Something we're working on with the NIH. In this case, the system, the pharmacy kiosk prototype, will dispense a plastic cassette. Inside the cassette will be both the dropper to collect the sample and the substrate. So you'll use the dropper from it. You'll put the dropper back, and you'll insert the whole cassette back into the machine. And the cassette is self-contained, so that there's no possibility of spilling the, in this case, saliva sample. And it is also sterilized prior to being deposited in a disposal bin. And the laser probe on the machine sits above the sample. So not only is the sample self-contained in the cassette, but it has no possibility of interfering with the laser probe.
Okay.
So these are some of the parameters, as you mentioned, for instance, contamination that we're working on for the pharmacy kiosk. And I'm happy to provide a copy of this presentation afterwards. And also, if you can provide me with a copy of the video, that would be excellent.
Absolutely.
Thank you. So the workflow is very straightforward. If we're using saliva, we're actually working on a version of the cartridge that you can lick, as opposed to needing a medicine dropper and to deposit sample. Our ideal use case would be to have the cartridge dispensed, licked, inserted back in the machine, because then you eliminate issues with correct sample volumes, you eliminate issues with ease of use, and various other advantages to doing it that way. So that's the next iteration we'd like to prototype, is a lickable substrate that can be inserted and tested immediately. So we'll get back with confirmation about that. But if we look at that potential use case, cartridge is dispensed, licked, reinserted into the machine, scanned for about 6 seconds, and results presented on screen. Because the system is run by a standard PC, they can be networked. These results can be reported to a practitioner. A practitioner can be streamed into the device. So you might want to have a consultation with a medical professional on screen. And also, the results can potentially be integrated into existing electronic health record systems.
Meaning that the results will be included into the electronic record, the electronic medical record of the patient?
Yes, that's possible.
And how do they have access to the physician? It's someone from your company, or it's their own physician?
So if we integrate with a, let's say a given hospital system is using, let's take an example of an EHR, Epic. Maybe a hospital system is using Epic. We would work with the hospital customer to integrate our software with Epic, so that the results would be controlled and disseminated in the customary way that they prefer to do things.
Okay.
If we want to look at just some of the studies that we've done to date, Harvard Beth Israel Deaconess Medical Center, High Core Lab at Lady Pohamba Hospital in Namibia, we mentioned these a little bit. In those two, we had a total of 307 subjects, 104 at Harvard and 203 in Namibia. And we achieved an overall accuracy of detecting coronavirus of 98.4%, a sensitivity of 99.6%, and a specificity of 98.1%. And these are our best stats to date.
Which was your gold standard for this detection of accuracy, sensitivity, and specificity?
Polymerase chain reaction.
Excuse me, PCR, yes.
PCR.
Yeah,
we always. Go ahead. I'm sorry.
Which test did you use from which company?
You know, that's a great question. I would have to get back to you on that. I'd have to reach out to Harvard and the folks in Namibia and get the manufacturer's name for you.
Okay.
So these stats are so far under ideal conditions. We've tested various sample types, and some are better than others. We can do it in all instances, but right now our top performance is 98.4% accuracy, 99.6% sensitivity, and 98.1% specificity.
And why in Namibia?
Great question. Really, 3 reasons. Reason number 1, cost. So, for instance, the Harvard project cost about $1 million, and took place over a year and a half, and culminated with 104 patient validation at the very end. The Namibia project cost 10% that amount. Took place in 2 weeks. So from a speed and cost standpoint, it's advantageous to work with emerging markets, but also population diversity. So we wanted to look at a much more diverse population than white people in Boston, and we found that the performance is equally high in both cases. And then finally, the privacy laws in the Health Information Patient Protection Privacy Act here in the United States. It makes it very difficult to obtain demographic and other information about patients that is easy to get in Namibia. So for that reason, we were interested in things like age, gender, confounding diseases, time since onset of symptoms, CT counts and viral loads, and all of these things were much easier to get in Namibia at low cost than it would be the case in the US.
So, you know, if you are looking for diversity, you have to come to Israel, as you know.
Please give me a reason. Yeah, I'd love to. I've been to Israel twice. I was on a fabulous trip in 2015. The Schusterman Foundation gave me a week-long tour of the whole country. They spared no expense. Everything was fantastic. And they asked me prior to the trip if I'd like to meet anybody in Israel, if I could pick one person who I would like to meet. And I said, I'd like to meet Shimon Peres. And they arranged for a private audience for our tour group with Shimon Peres, which was fantastic. So former president and prime minister and Nobel laureate. And so I was very fortunate to have that experience in Israel. I'd love to go back. My wife has never been there. So I look forward to opportunities to collaborate.
Yeah, Irene, I reached out to the Ministry of Health here in Israel and talking to a group within the ministry called Healthcare Israel, and their CTO is considering how they can possibly start a study here in Israel, and I should hear from them soon. So we have reached out to them, and and hopefully they'll move forward.
Okay.
Yeah, one of the top professors at Sheba Tel HaShomer Hospital is an advisor to the ministry, and he's part of the group that's now analyzing and checking to see how this can move forward.
Who is it?
The Keller is his name.
Okay.
Professor Keller, if you know the name. You know the person?
No, but in fact, perhaps you have to be in contact with Sheba ARC. You know, they have a center for innovation, and they are doing a lot of clinical trials with their data. So you have to be in contact with Eyal Zimnichman. So I will give you the contact.
Okay, great. Yeah, thank you.
I was a little bit late, so I would like to know what is the range of your wavelength in Raman, and do you need very powerful Raman you use?
Great questions. Our first generation product uses off-the-shelf spectrometer technology that is widely available at 785 nanometers, and is high performance, but almost a consumer technology at this point. So, for instance, there is an Israeli company called Consumer Physics, and they've developed a handheld 785 nanometer Raman analyzer that is good enough for doing things like determining macronutrient composition of animal feed, like how much water, how much carbohydrate, how much fat, things like that. Our system is on the high performance end of commercially available 785 nanometer spectrometers, but the technology is so mature that it ships in a standard cardboard box with solid state components, much as you would ship a laptop. Although it's more expensive than a laptop, obviously. So we do have a proprietary, a patented next generation spectrometer system that will be an estimated 2,000 to 4,000 times improvement in spectral resolution over current technology. And that one is very exciting in terms of what we'll be able to do that competitors simply won't be able to do in the future. So, for instance, right now we may be looking at millions of molecular artifacts, but with the next generation system, we'll be looking at billions or trillions of them, and we'll be measuring them with much higher precision than has been possible before. So you'll be able to answer all sorts of interesting questions about patients in the future using the next generation shorter wavelength technology. So, for instance, you might be able to say this person has coronavirus, but it's also a severe case of a specific variant, and they are also a smoker. So we're excited. We're excited to implement that new system. But we wanted to start with off-the-shelf technology because we can make it more cost-effective and reliable in the short term that way. And we augment that technology with our proprietary nanoparticle substrates and machine learning methods and other items.
Can you test several samples at the same time of different saliva?
It can be done. So we have been in touch with a high throughput screening company from the Netherlands. And the concept there is to position multiple laser probes above a 96 well plate and have them scan multiple samples at a time in succession across the plate. It's feasible to do that. We've not prototyped it yet, but we've looked into it. And for training the machine in the future, that's very exciting, because if you could scan, 100 patients, in a matter of minutes and upload that training data, and then very, very quickly have a classifier for a new disease. So a short answer, yes, but we haven't done it yet.
Thank you.
Okay. We work with some great folks. I'm a serial life science entrepreneur. I sold my last company for $22 million to a publicly traded firm. Great machine learning developers, great scientists. I won't belabor the point here on those, but highly accomplished. We also work with an excellent regulatory strategist who's been responsible for 30 plus medical device approvals in the United States, Europe, and Japan. And we're fortunate to have an incredible board of directors, including someone who started a health information technology company, a simulation software for the Defense Advanced Research Projects Agency, and sold his company for $25 million to a firm in Singapore. So there they are. We are now partnered with a variety of groups. We are partnered with Ocean Insight, a manufacturer of high-performance spectrometers. We have just done a deal with a large defense contractor in the U.S. They sold 1.5 billion needles and syringes to the U.S. government during the pandemic. That's 187 Boeing 747 jets full. And we're working on now, thanks to David Lidoff and Arya and Tatum, we're working on a partnership potentially with SciMed Global about doing a 5-disease study on saliva at the Liverpool School of Tropical Medicine. That has not been formalized yet, but we're working on it. Those are the highlights I have for you today. I would love to show you the machine, and I can show you how it works. Would you like that?
Of course.
Sure.
Okay, great. So we can go ahead and close this, and then I'm just going to walk you over. I better put on my gear here. And let's get some gloves.
Do we need a mask?
Because he is
if
in
you
a
like.
hospital. You're in a hospital, yes?
Yes, I'm in Johns Hopkins right now.
I wanted to ask you a question. Is it possible to do that on many other fluids like urine, blood? Blood you have already answered, but or any kind of fluid? Or
Yes.
okay.
Yes, any fluid we can currently use on the current system. Some, there are advantages and drawbacks to different fluids for different diseases, and also how easy it is to characterize a given fluid. So transparent fluids are very easy to characterize. Opaque fluids take a little more effort, but yes, we can do them all.
Okay.
So here we have the biosafety cabinet. And I'm going to show you this. This is the machine here. It's fairly small. It's a little bit smaller than a microwave oven. It can be carried. It has a handle in the back, and you can carry it like a briefcase. And here we've got a laptop set up with our software application, which is very simple. And I'm going to show you. Oops, I need to turn on the spectrometer. Pardon me. I'm going to show you how easy it is to program the system to be able to diagnose a new disease. So if we open the software application, let's see, can I position this so that you can still see everything? Are you able to see the equipment?
Yes.
Yes.
Okay, wonderful. So what I would do in this case, I would simply select one of our proprietary nanoparticle coated microscope slides. And in this case, let's take a sample. We have some coronavirus positives here. So this is training mode for the software. And all you have to do is type in the sample number. Excuse me 1 minute. Do you know which sample we left off on? Let's see. Can you tell me which was the last sample number? Since this is actual training data, I just want to make sure that we input the information correctly.
So you're gonna do the next one?
Next one, please.
So COVID,
Okay, so we're going to do coronavirus.
John Hopkins,
Yep. And JH for John Hopkins.
291.
And this is sample number 291. And we're going to select the classification of this sample. This will be an infected person, so we're going to select positive. We'll get some
A1.
sample here. All you have to do is place the sample on the microscope slide. Insert the slide into the machine. Machine is on, and click scan. And you can see that in training mode, you can get an actual, you can see what the laser spectrum of a COVID infected patient looks like. And it takes about a minute to collect data on a new patient. And it takes about 6 seconds when you run a test.
We don't see very well your screen.
The screen, okay. Let me see if I can make that a bit bigger for you. So I'm gonna open up the software application again. This is training mode. So in training mode, you enter the patient identifier. You select from the drop-down menu whether this is a healthy or infected patient, and then you click scan, and it performs a series of laser scans of the patient. It charts their molecular spectra, and then that data goes into a repository where predictions can be made based off of it. And when you open the software in testing mode, I can show you that as well. It's even simpler. We've designed it to be a very intuitive user interface. And in testing mode, all you have to do is enter the patient identifier here, click continue. It will scan for a minimum of 6 seconds, and the result will be presented on screen. It's a very, very simple process. Any questions about that?
No, it's very impressive.
Thank you. Okay, well, I think that's all I have for you today.
If we want, for instance, if there was a war and we want to do that at high speed, how much money it can cost?
How much does the system cost? Well, right
Yes.
now, building them one at a time, it costs us about $28,000 per machine. However, you know, there are more affordable components that are available, and the cost comes down with scale. So the more we make, the cheaper the machines are, and we are always.
It should.
Yeah, yeah.
It's not very difficult for physician.
Yeah.
In medical terms, perhaps more difficult, but in physical terms, it's not very difficult, I think.
That's correct. And we are always happy to support our allied nations in their biosecurity efforts, and we do have the ability to do that through our U.S. government contacts.
And so in your apparatus, you have a, let us say, physical unit, you might say, generating and capturing the data. And then you have the software to interpret, to analyze the data.
Right.
So could you tell us a bit more about the software, because this is really where the AI part lies and where you benefit from the machine learning, I presume.
You would like to know how the software programs itself, or?
No, just to know a bit more about the data interpretation, because of course this is really I would say where there is a lot of added value to your technology, because this is the part that will lead that will transform data to decision making, right? Which I presume also in your certification process will be something that will be clearly scrutinized by the agencies.
Sure. I am not able to disclose a whole lot about how the software works. It is different from and better than the techniques that are traditionally used in universities for Raman disease diagnosis. So, for instance, a simple method that's been used in the academic literature, a simple machine learning method, is called principal component analysis. Other examples are linear discriminant analysis, random forest. We do not use those techniques. We use a more advanced and proprietary method, which is both results in more accurate predictions and is also faster to train. So we have a method, we have a machine learning method that, depending on the size of the training set, and we believe we need a minimum of 36 infected patients to reach 99% of possible sensitivity. The classifier or the diagnostic test for that particular disease, can be generated by the software in between 1 and 5 hours.
Okay.
If I understand well, let's say that suddenly we have a new pandemic, and within a few days you have more than 36 patients. And suddenly you see something new that you cannot say that it's coronavirus or whatever virus, new virus is it, but you are able to raise an alert to say, hey, we have something new going on in many patients in a very small time, short time. Is it the idea?
Yes, less than a day. So we can, if you give me 36 patient samples, based on our current estimates, we can generate a new diagnostic test for that disease in less than a day. So that same day that you provide us with samples, we can generate a new diagnostic test, and we can disseminate it worldwide, the following day.
But you will not be able to say that it's this or this virus.
Well, we may or may not be able to. So if it's a new thing and we don't know what it is, then we can generate a diagnostic test for disease X.
Okay.
if it's similar enough to something that we've already characterized, we may be able to say, well, this is a flu variant, or this is a coronavirus variant. We may be able to say it looks very similar to a previous coronavirus,
Okay.
and we might be able to characterize it that way.
And when do you think that you will receive an agreement from FDA?
Well, I hate to predict when the government will will do anything, because they are not accountable to me. Optimistically, a year. If everything goes well, a year, I would say, but probably longer than that, just because of the typical pace of the regulation.
And actually, in line with this, the previous question, how do you actually are approaching this, convincing the agency that your bioassay is really accurate, and because again, I think I'm very impressed by the power, and I would say the versatility as well of your methodology. But then you mentioned this part about we don't need to know which biomolecule we are really documenting as being within the saliva, which again, I'm not a specialist in diagnostic, but which I would react to as being quite unconventional. So
Right.
you have to convince that you have a great method, albeit unconventional, speaking about regulatory agencies with people who are really thinking in a very, very conventional and codified manner. So what is your approach? How are you going to convince those guys?
Sure. So we expect the initial approval to be a challenge, and we are engaging with FDA and NIH stakeholders early and often. And ultimately, they will define for us what is required. One strategy that is possible for achieving approval in the shortest amount of time would be we could submit for classification as a diagnostic aid, in which case there would still be a medical professional in the loop. And the medical professional would collect the patient symptom status, then run the machine, use the machine and the symptom status to inform a medical diagnosis. That may be the lowest bar for us, but we're still working with FDA to determine.
I think it makes sense, because if you come with an intelligent machine helping human in decision making is one part. If you say, we want to replace a human expert, it's another level, because there is the part about liability, right? Which is another, But my question was not exactly that. Classically, when you come with a new method, you have to compare the outputs with what conventional diagnostic methods will yield. In fact, you had a part on this with respect to comparing with PCR for COVID, for example, diagnosis. So that's, I guess this is, I would presume from the standpoint of you say it's the IRB, right? IRB is a dedicated body from the FDA regulating diagnostic. Am I correct?
So an IRB is an institutional review board at the level of the academic institution that approves the parameters of a study to take place at their university.
Okay, I'm sorry. All right. So I presume that the FDA or the regulatory body will ask to compare the predictive performance of your machine with what they know. Those guys, the way they think, right? And I would say rightfully so. So that means for you, presumably, working on the number of samples, as you say, positive, negative controls, and comparing with standard methods for diagnosis of a given disease. I presume that's
right.
That's the part. Sorry, good.
We always compare to PCR. We always compare to the gold.
Okay. And I think that's a great approach, because it's very PCR is actually specific, as you said, of a given gene or molecule, and well obviously well accepted. Now, I was thinking, I think again, I think it's a great approach, this Raman spectroscopy approach. You may also even in your development phase consider adding to your sample a reagent, for example, in the form of a monoclonal antibody or monoclonal antibodies, to see if you have a sample without those reagents versus a sample where you add up those reagents.
Sure.
If you have an antibody recognizing, a biomolecule, you will have, immune complexes, which of course will translate into a specific signal in your signature and your spectra, and you may well be able to identify some of the molecules that you detect with your method, which I think would be a fairly easy, I would say, addition to the proof of evidence that you already are generating.
Yeah, you're absolutely right. That is something we will do. Contrived samples featuring various concentrations of
a given molecule. Okay.
Biomolecules and substrates.
Yeah.
Anyway, you have complex samples. For example, in our hospital, we have a lot of patients with cystic fibrosis. So usually they have Pseudomonas and other pathogens. So do you think there is no problem with some interferences or something else?
We don't have any indication of limitations on what kind of disease states we can program for. So we're thrilled to explore those. We started with infectious disease because that's where the urgent need and the funding was. But we are looking at different classes of disease, and we are interested in markers of all kinds. So we're excited to collaborate with researchers who have a unique focus. So if your focus is cystic fibrosis, we'd be thrilled to set up a study to determine whether or not we can accurately detect cystic fibrosis.
Okay.
Can you apply your technology to cells as opposed to soluble molecules? I mean,
sure.
Well, you detect viruses and bacteria. Perhaps you can also characterize spectra coming from cells.
I think it's highly likely.
Okay.
And there are examples of Raman spectroscopy being used in tissue pathology.
Okay. Which then you can apply to cancer or stuff like that. That would be great.
Yep.
Yeah. Okay. Very impressive.
Thank you.
Mm-hmm.
Very impressive.
Thank you.
When during the COVID in France, in the pharmacy, the role of the pharmacist has been really very much enhanced because they were suddenly involved in the vaccination and in the tests. So I think that could be also, at least in France, could be something that will be very welcome by pharmacists. I don't know if you can say that, Philippe, and all of you, but to be an interface with the patients and to perform this very easy diagnosis in the pharmacy, it's very quick and very easy access, and will be a very good role for the pharmacist. I don't know in other countries.
I think so. I mean, that's sort of our vision, is that the pharmacist will now become the primary initial point of care for patients, as opposed to the urgent care center or the emergency room or the traditional doctor's office. So, for instance, in the United States, on average, it takes about 18 days to get a doctor's appointment. And even if you go to an urgent care clinic, it's very expensive, and you will wait hours to be seen. In this case, the system can be at the pharmacy, and the test can be performed there, in minutes, in seconds to minutes, and the pharmacist, as a more central player in the healthcare chain, and potentially capturing more of the healthcare revenue at the pharmacy, is something that I believe will happen. So some of that upstream clinical revenue is going to now move to the pharmacy, and the pharmacist is going to become a more important figure in the process of how ordinary people access care in the future.
I think if you create value for healthcare professionals, those guys will help you to promote your technology for sure.
Sure.
No,
Yeah,
that's
we would
another.
love to. We'd love to have a panel of key opinion leaders.
Sure.
And what
In
about,
hospitals, yeah. Sure.
what about coagulation test?
Can you?
Coagulation test.
Relation test. Can you clarify that for Dave?
No, coagulation.
Coagulation.
Oh,
yes.
coagulation.
When you have clot, or not.
You know, again, not something we've done, but my hunch is that it would be easier than detecting a virus. Not something we've tried, but you know, again, anything that you're interested in testing for, you know, we're happy to collaborate on. And our priorities have been infectious disease as a result of recent history, but we're excited to assess the technology for all different other pathologies, and if that's an area of interest to you, we're thrilled to explore it. I am going to need to go relatively soon so that I can finish up testing for the day, and then take a few more meetings. So if there are other questions I can answer in the next few minutes, that would be great.
Sure. I can actually translate a question we have from one of our colleagues, that this is this type of methodology, as it appears, has been used on hemocultures from patients with bacterial infections to try to detect enzymes associated with bacterial resistance to antibiotics. Although, as I understand, from the question, in this case, the molecule or the biomolecule which was analyzed or detected was known. It was not a black box, so to speak. I don't. Are you aware of that?
I'm not familiar with that particular line of research. I know that sepsis is an urgent and very important use for the technology, but I've not heard it used for antibiotic resistance until now, and that's very interesting. Thank you for bringing that up. If you can approach developing a new test from either direction. The easiest and fastest way is to let it characterize whichever molecules it thinks are relevant. But if you prefer to start with a known molecule, thoroughly characterize that, and then create contrived samples at various concentrations of that molecule in biological material, it can be done that way as well. So very interesting that it's been done for antibiotic resistance. Thank you for pointing that out.
Okay. Do we have perhaps one last question, if any?
I just wanted to tell you, David, that both David, that this will be on the channel, YouTube channel of the Academy. We will circulate this record to all the group and also in the Academy. And we will send you the link so you can use it as you want. And thank you very much for your time and your explanation. It was really very, very
very clear, very exciting.
Thank you very much.
Well, it's been an honor to be here. I hope to see you in Paris and in Israel. And thank you so much for allowing me to present to this illustrious group. And I look forward to more in the future.
Thank you.
Okay.
Thank you.
Thank you very much.
So
Have
much.
a nice evening.
Thank
Or nice
you.
morning.
Thank you. Bye-bye.